You can see my screen, students.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay, so a little bit of brush up. I will not go into the contributions of Anthony Van Leeuwenhoek because we are not continuing with that. I will just brush up as to what Robert Koch has done. Robert Koch, he is founder of modern bacteriology, and he is also called as father of microbial techniques. This is what we had studied. He also explained the causative agent of anthrax, that is Bacillus anthracis. Again, details we have done in the previous class, so I will not go into the details of that. He also, which is a bacteria, he also told the causative agent of tuberculosis, which is Mycobacterium tuberculosis. He explained how it spreads, which organism it is, and for which he was awarded the Nobel Prize. After that, he again explained about the causative agent of cholera, that is Vibrio cholerae. Now all these are in the field of bacteria. Okay, so that is why we had seen that he's called as the father of he was considered in bacteriology. Then he also explained to us as to how to observe the bacteria under the microscope, something which is called as slide preparation, which includes smearing of the organism, that is smear making, then fixing the organism to the slide, that is called as heat fixing, and also since the microorganisms are colorless, he also taught us how to use aniline dyes, or he used aniline dyes so that we can observe the organism under the microscope. Basically, he gave color to the bacteria. So when the color was given, this is how the background, this is how the bacteria would look. So what did he do? This is one of the technique used in the microbiology lab, that is slide preparation, which includes smear preparation, heat fixing, and adding of color, use of aniline dyes. That is what he had done. Okay, then we went on to start about culturing, and then we stopped. Okay, we will do that in detail today. He taught us how to culture the organism or culture the bacteria in the lab. He told us that probably we can prepare media. There's something like how we make food for us to eat, for us to get energy. Similarly, he taught us we can prepare media, that is a food for the microorganisms. We can prepare media by taking water, adding different ingredients into water, and that is called as media. We can grow organisms in the media. That was called as broth, the liquid culture. The liquid media is called as broth. Okay, but we also saw in the previous class that liquid culture had certain disadvantages. Okay, when he was working with in his lab, there was a cut cooked potato which he left aside, and he saw that there was some growth in that. When he observed, he could find that there was organisms, and he could, looking at the cut cooked potato, he could find out, he could differentiate that there are different kinds of organisms, but that could not be happened using a liquid media. So he thought, yes, solid media is beneficial. We could separate out, we could find out what are the different types of organisms. And if I want to pick up one particular organism, that also I could do using a solid media. So that was the first solid media he used. Then one of the co-workers' wife, what she was, when she was cooking, she would use gelatin to solidify certain substances like your jellies, your ice creams, etc. So the same concept he used even for media, because he wanted now to solidify the media. He understood solidified media had advantages, which we have seen in the previous class. I just now also told you. So what he did was, he took the media, broth. Okay, now liquid media means I'm considering broth. He took the broth, and to the broth gelatin was added. Now when gelatin is added to the broth, it could solidify, just like how you use gelatin for preparing jellies and your ice creams. The same way it would solidify the media. Now the solidified media had those advantages. The media would get solidified, and if organisms were grown on the plate, he could able to differentiate, are there different types of organisms, or is there only one kind of organisms. He could also pick up one kind of organisms. So he used gelatin for solidification of the media. Okay, is that clear, students?
Yes, ma'am.
Yes, ma'am.
Okay. Now, how much of gelatin was used? That is the next question. 2%. Again, textbooks gives you variations of that 1.5 to 2%. Now, generally, we can stick on to 2%. Okay. 2% of gelatin was used. Now, what do you mean by 2%? That's 2 gram of gelatin was added in 100 ml of water. So, that's how can say. So, 2% of gelatin was used. Now, gelatin had certain disadvantages. Now, why did he add gelatin? Basically to solidify the media, isn't it? Now, gelatin also had certain disadvantages, so then he had to move on to another component to solidify the media, that is agar. Okay, now what are the disadvantages? Can anybody tell me what are disadvantages do gelatin have? You prepare gelatin and then jelly at home, ice creams at home using gelatin. What disadvantages are there using gelatin? If you go to kitchen only, no?
Ma'am, on like if it
Okay.
we have like
Tell me.
keep it on low temperature to again solidify it.
Yes, very good. So gelatin, it had certain disadvantages like above a particular temperature. That is, if you increase the temperature of gelatin, the it would not remain solid. It would start liquefying. Okay, above 25 degrees centigrade. If you increase the temperature, the gelatin would melt. Like your ice cream, you keep ice cream in the refrigerator, it will remain solid. But then you keep it long outside, it will start melting. Even at room temperature, it will melt. Isn't it? The same thing happened with gelatin also. What nutrient gelatin? If I have to take an example of that, I wanted a solid media. Even at room temperature, it should remain solid, isn't it? But if the temperature increases above 25, what would happen is the gelatin would start liquefying. The solid media, which wasn't solid at 25 or below 25, now would start becoming liquid. Okay, so that should not happen. That was one of the disadvantages of gelatin. One more thing is that it serves as a nutrition for some of the microorganisms, which means not all, some microorganisms, they have a particular enzyme within themselves that is called as gelatinase. Okay, they have the capacity to utilize this gelatin. So means, in other words, if I have to say, consume the gelatin or eat up this gelatin. So if they start consuming the gelatin, will the gelatin remain in the media? Definitely no, it will get used up. So again the media would start liquefying. Okay, one more thing is, bacteria to grow requires 37 degrees centigrade, and 37 definitely the gelatin will start liquefying. So gelatin was not used. Initially gelatin was used as a solidifying agent, but these are the reasons why now gelatin is not used. Okay, it was then replaced by another component, which is agar. Okay, details of agar you would be studying when you're studying about culture media. Okay, that is a different topic as such, but then you should just know how did it move on. What was the first solid media, then what was replaced, and then what is used now. Agar is one component which is prepared from seaweeds, and that does not have any what do you say, any nutritional factor, and that remains solid up to 50 degrees centigrade. Okay, so happily even at room temperature it still remains solid, and no organisms can utilize agar. And that is why agar is still used as a solidifying agent. Around 2% of agar is used. Is that clear, students?
Yes, ma'am.
Yes,
Yes, ma'am. Yes,
agar
ma'am.
is
Okay, so initially it was cut cooked potato, it was broth, that's liquid media. It had certain disadvantages, moved on to solid media, that was cut cooked potato. From there we moved on to gelatin had certain disadvantages, then it went on to agar, which is a solidifying agent, which is still used now. Fine, moving on to the other concepts.
agar is prepared from
Agar is prepared from seaweeds. Other things, what were his other contributions? He has one major contribution which I'll talk to you a little later. Another contribution which he has done is that he also observed the microorganisms, studied the organisms that is present in soil. He said that organisms are present in soil, it is present in water. He also told or compared the relation between the organisms that is present in water and air. Okay, that is present in soil, water, and air. Okay, how are they related? That all these studies he has done. Basically, you can say in bacteria. Okay, another important thing, that is one other contribution of his. Next, he explained or he told about sterilization. He told or he introduced, I told you about sterilization, isn't it? It's killing of all the organisms that is present, irrespective of whether they are resistant or whether they are in the spore form or whether they are in the which vegetative form, whichever. Killing of all the organism is called as sterilization. Now, this concept was introduced by him. He told that we can use one instrument called as hot air oven. We can use the instrument called as hot air oven for the killing of the microorganisms. Now again, I will be explaining to you what is the concept of hot air oven, but then he told this is one instrument. He designed this instrument, he explained about this instrument. This instrument can be used to kill all the microorganisms. Is that clear?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am. It was he. This was his another contribution. It was he who told that he told about antiseptics. Okay, he gave the difference. He distinguished between what is the meaning of bacteriostatic, bactericidal. Okay, when if you increase or decrease concentration, bacteriostatic can act as bactericidal. That is explained or told by Robert Koch. Okay, so is this slide clear? Can we move on to the next slide?
Yes, ma'am.
Okay, next, very important thing. Okay, he told something called as germ theory disease, or he explained germ theory disease. What disease? He told that if there is any diseases, it is because of the presence of microorganisms. That is called as germ theory disease. Okay, that's called as
Ma'am,
germ
can you
theory.
please repeat?
Yeah, Koch. Okay, Robert Koch. He told about the germ theory disease. Okay, now what is this germ theory disease? He told that if there is any disease is associated to the presence of germs. Germs means microorganisms. Okay, if anybody is suffering from any disease, that is because of the presence of germs. Germs means microorganisms. That is called as germ theory disease. Okay, now having this relation of disease, okay, with the microorganism. Okay, and also remember he had told about the etiology of cholera and tuberculosis. Keeping all these into consideration, he proposed certain postulates. Okay, now those postulates are called as Koch's postulates. Okay, it was he started working on that in the year 1884, and the Koch's postulates was told in the year 1819. Now what are those Koch's postulates based on the germ theory disease? We will be studying now. Okay, now what are the Koch's postulates? Let us see. Four postulates he gave. Okay, four theories he gave based on the germ theory disease. Now what are the Koch's postulates? The first postulate he told was, now if any individual, animal or human being is suffering from a disease, okay, it there will be definitely presence of microorganisms in that person. Okay, only then that is called as a first postulate, which means, in other words, I will tell you, if any person is suffering from a disease, any animal or any human being is suffering from from a disease, it is because of the presence of microorganisms. Other words, if I try to remove, if I try to observe the blood sample from a diseased individual, I should be able to see the presence of microorganisms. It is because of the presence of microorganism only the person is having a disease. Okay, so that is the first postulate. Did you understand?
Yes.
Yeah. Okay. Suppose I take the blood sample from a healthy person or a healthy animal, I will not be able to see, I should not be able to see any microorganism. So that is Koch's first postulate. First postulate means if anybody is suffering from a disease, that is because of the presence of microorganism. Okay. So that is first postulate. The second postulate is, now I told you the first postulate says that any person is suffering from a disease, that is because of the presence of microorganism. Now that microorganism should be able to be grown or should be able to be cultured in the laboratory conditions. Okay, this is what I have shown. This is the media, this is the culture media that I have shown here in the picture here. Okay, the organism from which the the person was suffering from, I should be able to see the microorganism in the blood sample, and that organism which I have seen under the microscope, I should be able to grow that microorganism in media. That is the second postulate. Did you understand?
Yes,
Yes,
ma'am.
ma'am.
Yes,
Yes,
ma'am.
ma'am.
If you see the healthy individual here, healthy person or healthy animal, there are no organisms. Okay, and definitely there are no organisms growing on the media. But whereas if you see here, if it is a diseased animal, from the diseased animal I was able to see the microorganisms, and those microorganisms I can grow. This white thing what I have shown is the growth of the microorganism. Able to culture the microorganism in the laboratory conditions using any media. Okay, so that is the second postulate. Moving on to the third postulate. Two postulates are clear.
Yes, ma'am.
Yes, ma'am.
Yes,
Moving on
ma'am.
to the says that if I take organisms are growing in this media, isn't it? Now if I pick up the organism from this plate and I inject it into a healthy animal, okay, the healthy animal also should have the same symptoms what the first animal had. That is the third postulate. Did you understand this postulate?
Yes, ma'am.
No? Confusing? No? Okay, see, let us start. The animal was diseased, isn't it? Not deceased, diseased. Now, I took out the blood sample, I got the organism. Okay, the organism, the person or the animal was suffering because of the presence of microorganism. So, it had certain symptoms. Microorganisms, first postulate. Those microorganisms I was able to grow in the laboratory conditions on the media. That is a second postulate. That microorganism I took and I introduced into a healthy animal. That healthy animal also got the same symptoms, okay, what the first animal had. Okay, so that is the third postulate. Third postulate is, if I take out the organism, the growing organism on the plate, and I introduce into a the animal, so should show the same, it should not show another symptom, it should show the same symptom what the first animal had. Okay, so that is the third postulate. Is it clear?
Yes, ma'am.
Yes, ma'am.
Yes,
Yes,
ma'am.
ma'am. Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Now that animal is anyway suffering, it is got the symptoms. Now suppose I take out the sample from this second animal who is suffering, who is diseased. Okay, I take out the sample, I should be able to see the same organism that was taken out. Then that I had seen when I had isolated the organism from the first animal. So which means this organism, this microorganism and this microorganism should be exactly the same. I should not be able to see another different kind of organism. The organism that is isolated from the second animal and the organism that was isolated from the first animal should be the same. Okay, which means both the animals were suffering from the same disease, was isolated. That is the fourth postulate. Did you understand?
Ma'am,
Yes, ma'am.
Yeah. Okay. Now quickly we'll see what are all the postulates. If any animal or any individual is animal is suffering from a disease, it is because of the presence of microorganisms. I should be able to see those microorganisms. That is the first postulate. Those microorganisms, I should be able to grow that in the culture media, the laboratory conditions. Now this organism who is growing on the laboratory condition in the plate, if inoculated or if injected into a healthy animal, that healthy animal also should have the same symptoms what the first animal had. Now once the second animal is getting the symptoms, okay, and I try to isolate or try to check which are those organisms. So those organisms should be the same as the first organism that I had isolated from the infected animal. Okay, so these are the postulates. These are called as Koch's postulates. So in the exam, if they ask you about Koch's postulate, you have to explain. You have to define the postulates and you have to explain those postulates. Okay, and mention about the germ theory disease. Clear? Clear?
Yes,
Hello?
ma'am. Yes, ma'am.
Yes, ma'am.
Sleeping or started
Yes, ma'am.
Ma'am, the fourth point is kind of not visible actually.
It's not visible.
Like identical to that's the only part I can see. After that, it's not visible.
Like the same organism. Okay, it's gone. I will send it to you. Okay.
Ma'am, identical to the chained
To
organism.
the same organism that was isolated from the first animal. Yes. So these were the Koch's postulates. You have to define these postulates and disease. Definitely have to mention about the germ theory disease here. Okay. So is this clear, students?
Yes. Yes, ma'am.
Yeah. Everybody. Any doubts?
Yes. Yes, ma'am.
Ma'am, yeah.
Ma'am, identical to the what you said.
Identical to the same organism that was isolated from the first animal.
Ma'am, could you just go back 2 slides? Ma'am, 2 slides behind.
What?
One more, ma'am. One more, ma'am. Okay, this one.
This one?
Yeah, ma'am. No, the other.
This one?
Next one. Yes, ma'am. Ma'am.
This one? Sterilization.
Ma'am. Yeah, ma'am. Yeah.
Okay. Anybody
Yeah, ma'am.
is okay.
Yeah, ma'am. You can change.
So these were the postulates. Students, if they are asking you in the exam, explain about, definitely one scientist will be asked for a 10-marker question. Side heading should be what was his contribution, and you have to underline that and you explain about the contribution. Like for example, anthrax. Okay, Bacillus anthracis, that will be your side heading, and you have to explain about what he has done in anthrax. Then Vibrio cholerae, then you have to explain about that. So each one you have to explain in detail with the side heading, the main thing there. Okay, so these were the contribution of Robert Koch. If we have to quickly see. Right. These were the contributions. This is the pictorial representation, another pictorial representation of the same postulates. This is the animal. If the animal is suffering from any disease, I take out, I should be able to see the organisms. The organisms should be able to be cultured in the lab conditions. If those organisms, if when grown on the lab conditions, if I inject it into a healthy animal, the healthy animal also should get the same symptoms like the first animal. And this animal, I take out the organism from the second animal, I should be able to see the same organism that was identified when I had isolated the organisms from the first animal. So this is a pictorial representation, another representation of the Koch's postulates. So whichever is easy, you can probably see. Thank you. Now, quickly, if we have to see all the contributions of Robert Koch, if quickly I'll brush up. Okay, what did he do? He is father of modern bacteriology because his major contributions were in the field of bacteria. So microbiology, because he told about the various techniques. Then he went on to tell about the causative agent of anthrax, about the causative agent of tuberculosis, that is Mycobacterium tuberculosis, explained about that. Vibrio cholerae, the causative agent of cholera, he explained. Then he told about the techniques, because he's called as a father of microbiological techniques. He told us a method of slide preparation, smear making, heat fixing, and the coloring of the organism using aniline dyes. Then he was the first person to tell that culturing of the bacteria possible. He used first solid media that he used was cut cooked potato. Then that had certain disadvantage. I mean, he used gelatin to solidify the broth, and gelatin had certain disadvantages that was further changed to lard. Next contribution was, he was the one who told about the organisms that are present in soil, air, water, relation between that. He told us about the instruments that can be used for sterilization, and also he told what is bactericidal, bacteriostatic, when bacteriostatic can become bactericidal. Then he also told about the germ theory disease, taking this consideration example into consideration, the cholera and tuberculosis. He framed, or he gave postulates. Okay, that is called as a Koch's postulates. We studied about those Koch's postulates. So that is what we had studied in today's class. Clear?
Yes, ma'am.
Yes, ma'am.
Yes,
So we
ma'am.
brushed
Yes,
up.
ma'am.
We studied
Yes,
about
ma'am.
science till now. Okay.
Um,
I will be sharing this this. So you don't have any doubts?